The Bavarian Health and Food Safety Authority reported the first case of coronavirus infection in Germany today.
Hello, are you in the lab?
Hi, yes I am.
Could you please check for the expression of ACE2 in your dataset? This is the receptor of the SARS-CoV-2 virus.
Oh, it looks like they're actually expressed by specific airway and alveolar cell types.
That's interesting. This is potentially mechanistic.
The Robert Koch Institute reported 281 new cases of infections with SARS-CoV-2.
The number of cases is steadily rising. Current health safety measures such as mask wearing.
The WHO is currently working with a team of epidemiologists to investigate the origins of the virus. Hi Professor, so I just had the introduction for the Biosafety 3 level laboratory today. Do we already know what kind of sample we would be using?
It would be nasal swabs. Do you think we can get cells from there?
Ooh, I don't know, but we'll just have to see, right?
Hello?
Hi! So from the data, we can actually see that critical cases of COVID-19 have more cell-to-cell interactions.
Very good. And it looks like immune cells have a strong inflammatory signature. This is very important. We should publish this.
Hello?
Hi, we are going to collaborate with cardiologists to investigate COVID-19 in patients who have hypertension. Would you be able to help?
Sure, but why are these patients interesting?
ACE2 is also important for regulating blood pressure, so drugs against hypertension could affect its expression and therefore also disease severity.
Hey, so it seems like patients treated against hypertension have no increased risk of getting COVID-19.
I agree, and according to the data, they should continue to take their prescribed drugs, since it might even reduce the risk of critical outcome.
Hey, I just talked to colleagues from the pediatric department and we were discussing why there are almost no COVID cases of children in the hospitals.
That's an interesting point. Will we collect nasal swabs from children?
Yes, and we might start soon.
Hey, the children dataset showed some interesting differences.
Yes, I looked into it. It seems like the children have a pre-activated immune system and might therefore be more efficient at clearing the virus.
Oh, could that explain the differences between children and adults?
Yes, indeed.
My name is Emanuel Weiler and I work at the Max Selberg Center for Molecular Medicine in the field of regulation of gene expression in virus infections. By using single-cell biology, we want to figure out why an infection with SARS-CoV-2 can be life-threatening in some cases and not much more than a cold in others. In our experiments, we have observed how strong the inflammatory reaction is to the virus, particularly in certain types of immune cells. This supports the hypothesis that severe COVID-19 is due to an overshooting reaction of our own immune system and can inform the corresponding therapeutic strategies.
Hello, I'm Tommaso, and here at the Proteome Dynamics Lab at the MDC, we're studying the effect of SARS-CoV-2 on macrophages. Since these cells of the immune system act both as frontline soldiers fighting infections, but they're also a bit like generals, telling other cells what they should do. So we took macrophages from healthy donors, we infected them with SARS-CoV-2, and then we measured the abundance of 7,000 different proteins of macrophages to see how the virus was affecting and changing these cells. We found out that SARS-CoV-2 makes the macrophages go a bit crazy, so that the macrophages tell other cells to produce too much of that gooey stuff that is around the lungs, or fibrotic tissue. And this kind of lung fibrosis is what then causes severe respiratory problems in COVID patients. We hope that knowing this will help in coming up with better therapeutic options for COVID in the near future. Thanks for your attention, and bye!
Hi, I'm Sophia. I'm a PhD student in the group of Leif Eriksander at the Charité. Already at the beginning of the pandemic, it became pretty clear that COVID-19 is a disease with many different clinical manifestations, and that the immune responses to the SARS-CoV-2 infection plays a critical role in how the disease progresses. In collaboration with many other research groups, we performed our study to understand the innate immune responses to the SARS-CoV-2 infection at different disease severities. We therefore analyzed the myeloid immune cells, especially the monocytes and neutrophils, in patients with a mild and a severe disease course, and looked at different time points after symptom onset. We used single-cell transcriptomics and proteomics techniques in 2 independent cohorts and saw differences in the immune cell composition in these patients. In mild COVID-19 patients, we saw activated monocytes, which were basically ready to defend the patient against the virus. And in contrast to that, we saw in patients with severe COVID monocytes and neutrophils that were dysfunctional, and saw a considerably higher number of neutrophils that were immature. And these cells contribute then to an immune response which is insufficient and ineffective in severe COVID-19 patients.
I'm David.
And I'm Pinkus.
And we've been working with COVID-19 vaccines in the team around Leif Sander and Florian Kurt here at the Charité Department of Infectious Diseases and Respiratory Medicine.
We all know that the elderly make up one of the largest groups at risk for severe COVID-19. However, they made up only a small fraction of participants in the vaccine trials. We investigated the immune response to COVID-19 vaccines in a cohort of elderly persons and compared them to younger healthcare workers. What we found was that the elderly not only exhibited significantly lower levels of SARS-CoV-2 specific antibodies and spike-reactive T cells, but that the response was also significantly delayed, and 1 in 10 elderly didn't produce reactive antibodies at all. Thus, a timely second vaccination for the elderly is necessary to elicit an adequate immune response to COVID-19 vaccines.
Additionally, when evidence of rare but severe adverse events in younger persons after the AstraZeneca vaccine came to light, the question came up whether they should receive their second vaccination with an mRNA vaccine, such as BioNTech or Moderna. We found that the so-called heterologous immunization was comparably well tolerated, and in addition, compared to the classical regimens, the heterologous immunization even showed better immunogenicity. Therefore, we concluded that a heterologous immunization might be a suitable vaccination strategy.
So, the new COVID-19 vaccines have been a game changer in this pandemic, and we're looking forward to continuing our research.
Hi, I'm Niklas from the German Multiple Sclerosis Registry and the University Medical Center of Rostock. Worldwide, multiple sclerosis, or MS, is the most common neurological autoimmune disease of the central nervous system in young adults. As vaccinations to prevent infectious diseases play an important role in these patients, who are often treated with immunomodulating drugs, fear of vaccination reactions and disease worsening after SARS-CoV-2 vaccination are key questions of MS patients. In light of the exclusion of MS patients from the pivotal trials, in May 2021, the German MS Society, in collaboration with the German MS Registry, launched a Germany-wide observational study of MS patients consisting of an online baseline survey and 3 follow-up surveys. By mid-August 2021, 2,300 patients participated in the baseline survey and 1,800 in the first follow-up. On the descriptive level, more than 60% of the patients reported at least 1 vaccination reaction, 5 to 6% reported new MS symptoms, and 10 to 12% reported worsened symptoms after vaccination. MS relapses occurred in 3 to 4% of patients after vaccination. A causal relationship between SARS-CoV-2 vaccination and disease worsening cannot be concluded from the data to date. Long-term data expected next year will shed more light on this. Thank you very much and goodbye.
Hi.
Hi.
We're Mommsen and
Jakob.
And we're two clinician scientists working at the Charité University Hospital, usually in the Department of Neurology and
Pediatric Neurology.
And we are very interested in antibodies.
And of course, we kept our interest also during the pandemic. But we focused it on the novel virus to understand how our immune system responds to this new pathogen.
And to this end, we selected 10 patients which were hospitalized at that time and identified and isolated over 600 different monoclonal antibodies. And together with colleagues from the Department of Virology and Infectious Diseases and Pulmonology, we looked at how these antibodies can bind and neutralize the virus.
And by studying this large set of antibodies, we actually identified one that had the most potent effects. And then we used this antibody and gave it to hamsters that were infected with the coronavirus. And we saw that it protected the animals from developing disease symptoms. And these promising findings we now want to follow up and develop a therapeutic that can also protect humans from disease severity. And essentially all of us from a further spread of the pandemic.
Hello, my name is Géraldine Noyer and I'm an immunologist at the Charité, Department of Infectious Diseases and Respiratory Medicine. Our research focuses on questions that cannot be studied in patients. Therefore, we collaborate with scientists from the Free University who, in response to the pandemic, established hamster models for COVID-19 and with experts in novel single-cell sequencing technologies from the MDC. Together, we succeeded in describing the so far uncharacterized immune responses of one of the most relevant animal models for COVID-19 and identified an unexpected role of endothelial cells in promoting earliest inflammation. We found that immunity in this model is highly comparable to human immune responses. We can now relate immune cell behavior to disease severity and are better equipped to probe for cell-specific effects of therapeutics and vaccination and thereby aid development of medical strategies.
The COVID-19 pandemic has posed a high-level threat leading to intense scientific activities around the world, including numerous regional, national, and international epidemiological surveys and registry studies. However, it is exactly this multiplicity of scientific efforts that threaten to create a dangerous segmentation of information, resulting in a delay or even preventing urgently needed scientific findings. Most of the datasets are not standardized, making it extremely difficult to compare or analyze COVID-19 data across studies, as well as using AI and machine learning algorithms. Data scientists spend most of their time working on data preparation and not on tasks like the actual development of machine learning algorithms. A lot of their time is spent collecting, cleaning, and reorganizing data, compared to a small percentage which is spent on machine learning algorithms. To make data more accessible to AI and machine learning and reduce data preparation, you need standardized and interoperable data. This will ensure that algorithms have a clear data structure and semantics to work with, ensuring the validity of results and creating trust in digital technologies. To encourage coordinated action and assurance of semantic and syntactic interoperability and common language of the data collected, the German Corona Consensus Dataset, short GECKO, was created, giving scientists a standardized dataset for COVID-19 research as a universal working basis and language. GECKO is based on the PA COVID-19 study, the WHO-ISARIC protocol, and the data concepts of the LEOS register. However, the uniqueness is that it was developed by a multidisciplinary expert board containing approximately 83 data concepts and 280 response options from different domains like demographics, vital signs, and symptoms. Most importantly, making use of the international standards and terminologies like SNOMED-CT, HL7-FIRE, LOINC, ICD-10, GM, and ATC. The Pioneer dataset has provided researchers with a universal approach to understanding clinical data. GECKO has already created momentum that can be accessed online at any time. used by projects within the COVID-19 research network of University Medicine, Netzwerk Universitätsmedizin, and Europe-wide within different projects. By not standing still and staying prepared for future pandemics, the team around the Gecko is currently working on further modules to accompany the dataset, making it even more interdisciplinary by harmonizing and streaming research.